Hi, I'm Demirtaş. I'm from Turkey and I studied at İzmir Science High School as a 10th grader. My project's name is a research about the toxicity of PLGA and Borax synthesis to temozolomide on glioblastoma cell cultures. In this research, temozolomide and borax were loaded to polylactic glycolic acid nanoparticles and indocyanine green dye. In this study, it was aimed to increase the effectiveness of boron-nurturing cancer therapy and chemotherapy in brain tumors with a dose of a drug with the boron content of borax, to provide the lower toxic effects at higher doses on healthy tissue by targeted drug delivery on glioblastoma cell cultures, and to provide effective dose on targeted tumor. Firstly, I would like to give some information about the terms and synthesis materials. Glioblastoma is known to be both one of the most common types of malignant brain tumors and one of the most difficult tumors to treat. The most effective treatment for glioblastoma is neurosurgery, but chemoradiotherapy is applied for weeks with temozolomide for the tumors, which cannot be removed by neurosurgery. So, it is decided to study glioblastoma since it's a serious disease and is difficult to treat. Temozolomide is a chemotherapy drug that is used in the treatment of this tumor. Temozolomide is more preferred in the treatment of glioblastoma since it can overcome the blood-brain barrier. PLGA was used as a carrier for drug release because PLGA is a biodegradable polymer that can be used in medical devices and drug delivery systems. It also has minimal toxicity and it is approved by the Food and Drug Administration. Targeted drug delivery is the integration of a drug into an area within the body, affecting only the target area without any effects on other structures. The natural antiseptic properties of borax have enabled the use of this compound in medicine. In this project, borax was used because approximately 73% of the boron reserves in the world are in Turkey, and the use of a boron compound compatible with the nanoconjugate in such a study will increase the applicability of the project in an economic way. ICT is a fluorescence dye that is approved by the FDA, used in cell culture, biomedical files, and surgical procedures. In the first stage of the synthesis, we dissolved 100 mg of temozolomide in 20 ml of pure water and then added 2.5 mg borax. We dissolved 10 mg of PLGA and 105 mg of propylene glycol in 2 ml of chloroform. We added the resulting solution to the first solution. We solved 100 micrograms of indocyanine green in 20 ml of acetone and added it to our previous solution, and we gained our nanoconjugate. We added 100 ml of ethanol in nanoconjugate, put it to a sonicator to form a homogeneous nanoconjugate, and make the DLS analysis. DLS is an analysis method to examine the size and stability of biomolecules and nanoparticles which have the size below the micrometer in the solution. For DLS measurement, 600 microliters were taken from the prepared sample. The measurement result is proportional to the cube root of the size, so it's larger than this SPM size as expected. The results of the DLS measurements show the homogeneous structure of nanoconjugate. After this process, cells were cultured in order to indicate the effectiveness of the drug by loading it to these cells. In a 75 ml flask, 11 ml of medium were prepared with the DMEM, antibiotic and serum mixture for the cells, and the cells were added to it. The cells were incubated in the carbon dioxide incubator to defrost and replicate by sticking it to the flask surface. After that, 10 microliters were taken from the cells and they were counted with the light microscope. Temozolomide PLGA nanoconjugate was added to the medium. After this process, fluorescence and SPM images of the cells were taken. It was understood from the dark spots of the fluorescence microscope images that temozolomide PLGA nanoconjugate covered the surface of the cell completely, surrounds the cells. In 3D SPM images of the glioblastoma cells, showing the cell surface containing nanoparticles, temozolomide PLGA nanoconjugate completely covers the surface of the cell membrane, and surface is very smooth. Lastly, the toxicity tests for both temozolomide solution and temozolomide PLGA nanoconjugate were made in different concentrations. According to the graph that showed the result of the toxicity test, it was concluded that the bioavailability of the temozolomide PLGA nanoconjugate for 24 hours was high and did not have serious toxic effects on the cells. As a result of all these studies, it is observed that the bioavailability of the temozolomide PLGA nanoconjugate is high. It does not have serious toxic effects on the cells. It can increase the efficacy of the targeted treatment in glioblastoma, and it's a suitable system for targeted drug delivery. Thank you for your attention.